Good morning, good afternoon, and good evening. I'm Irfan Memon, a Regulatory Health Project Manager in the Office of Research and Standards, Office of Generic Drugs at FDA. I'm honored to be serving as your moderator today. On behalf of the Office of Generic Drugs and our colleagues across CEDAR, thank you for joining us for this year's Advancing Generic Drug Development, Translating Science to Approval workshop. Yesterday, we explored insights to product-specific guidance for complex product, from research to standards, and novel bioequivalence study design recommendation. As always, our goal is to move from scientific insight to regulatory clarity so that generic developers worldwide can advance safe, effective, and affordable medicines to patients more efficiently. Today, we'll continue that dialogue by focusing on two important themes. First, common deficiencies and resolutions for complex drug substances, complex critical excipients, and complex products. And second, nitrosamines, known issues and practical advice. Before we begin, a few reminders. You're invited to submit questions via MS Teams by using Q&A functionality, or if you are unable to use Teams Q&A functionality, please submit your question via email to ors@fda.hhs.gov with a subject line of AGDD 2025. When submitting a question, include the name of the speaker and session it relates to, as this will help us to route it to the right person. Please take a screenshot of this slide and submit your questions as early as possible and do not wait. Our team will be actively monitoring all questions submitted, and we'll do our best to address as many questions as possible during the designated Q&A sessions. All sessions are being recorded. The recording and presentation slides will be posted on the FDA website. Now, we are excited to kick off today's session with Session 3, Common Deficiencies and Resolutions for Complex Drug Substances, Critical Excipients, and Complex Products, which will be introduced by Dr. Yan Wang. You may remember Dr. Wang from yesterday, where she provided our Day 1 closing remarks. As Deputy Director in the Division of Therapeutics Performance 1, within the Office of Research and Standards at the Office of Generic Drug, Dr. Wang leads effort to advance pre-application development and regulatory science research for complex drugs products. Please join me in welcoming Dr. Wang to introduce our Session 3.
Thank you, Irfan, for the kind introduction. Good morning, good afternoon, good evening, everybody. On behalf of my session co-lead, Dr. Dominic Russell, who is a supervisory chemist in the Office of Pharmaceutical Quality, welcome to Section 3, Common Deficiencies and Resolutions, Complex Drug Substances, Complex Critical Excipients, and Complex Products. As we evolve in our scientific understanding, we have also seen an increase in the complexity of drug products entering the generic pipeline. Today's generic developers are tackling products that would have been considered impossible to develop maybe just a decade ago. Complex drug substances with intricate molecular structure and critical excipients that play active roles in drug performance, as well as sophisticated delivery system, all present very unique challenges that require nuanced approaches to demonstrate bioequivalence and ensure product quality. So what will make this session particularly valuable is its focus on the real-world problem solving. Rather than just the theoretical discussions, today's session will discuss actual deficiencies that have emerged during assessment process and the collaborative solutions that have been developed. This represents the collaborative learning from our research efforts, assessment of applications, which are distilled into actionable insights that can help you to avoid some common pitfalls and accelerate your generic development. Our first presentation on compositional sameness for complex polymeric excipients, progress and remaining challenges will be delivered by Dr. Bing Ching, Senior Staff Fellow in the Division of Therapeutic Performance 1, Office of Research and Standards, Office of Generic Drugs. Dr. Qing will address the ongoing challenges in establishing compositional sameness for these complex materials and related products. Following that, we'll dive into a very interesting case study with navigating complexity, key considerations in developing the oral semaglutide product-specific guidance. This presentation will be jointly delivered by Dr. Sammy Nazo, Senior Pharmacologist in the Office of Research and Standards, Office of Generic Drugs, and Dr. Jinghui Zhang, Senior Pharmaceutical Scientist in the Office of Pharmaceutical Quality Research, OPQ. Their collaborative approach will provide insights into the multidisciplinary considerations required for this complex product, enable the product-specific guidance development. Next, we will examine two compelling cases studies on Glatiramer acetate substance injectable and Pentosan polysulfate oral capsules, presented by Doctors Maotang Zhou and Keduo Qian, who are from the Office of Pharmaceutical Quality. Dr. Maotang is the Division Director for Division of Product Quality, and Dr. Qian is a chemist in the Division of Product Quality Assessment. These case studies will illustrate the practical application of regulatory science to tackle complex drug substances. Finally, and last but not least, Dr. Dahui Liu, Senior Pharmaceutical Quality Assessor in the Division of Product Quality Assessment for OPQ, will present on comparative physical characterization of iron products, addressing the unique challenges associated with the iron colloid in organic products. So as Irfan already mentioned, after our presentations, we'll have interactive Q&A panel featuring all our speakers. And please submit your questions, as this is your opportunity to engage directly with this subject matter expert. With that, without further ado, I'll pass the floor to Dr. Qing to kick off today's session. Thank you.
Thank you for the introduction. Good morning, everyone. Good evening and good afternoon. In today's presentation, my talk will be focusing on compositional sameness for complex polymeric excipients. In these presentations, I will first talk about what are the requirements for compositional sameness, and then followed by case studies about what type of information are needed to demonstrate compositional sameness for complex polymeric excipients. When is compositional sameness needed? According to the regulations, certain generic products need to be have the same composition as the ILD. So the regulation requires that generic drugs with parenteral, ophthalmic, and otic route of administration contain the same inactive ingredients in the same concentration as the reference list drug. Here, the same inactive ingredient is often referred as qualitative sameness, which is Q1 the same, and in the same concentration is often referred as quantitatively sameness, which is referred as Q2 the same. In addition to the regulation requirement, there are some other scenarios that demonstration of compositional sameness is required. For example, the PSG recommendations. Here, I just want to show one of the PSG on levonorgestrel intrauterine systems. In this PSG, although this drug products, it's not an parenteral, otic, or ophthalmic products, the PSG does recommend that a test product should contain no difference in inactive ingredients or in other aspects of the formulation relative to the RLD products may significantly affect the local or systemic availability of of active ingredients. Here, Q1, Q2 sameness is one way to satisfy the no difference in active ingredients. When for products that regulation require Q1, Q2 sameness, or the PSG recommend Q1, Q2 sameness, the applicant can submit control correspondences to the agency to request for formulation evaluation. In these cases, the agency often first evaluate whether the test formulation composition is Q1 the same. If the Q1 sameness is confirmed, the agency will evaluate whether it's Q2 the same. In the case of polymeric components in the formulations, the evaluation is very similar to other excipients. In most of the cases, are relatively straightforward. Here, just want to show some of the cases that are relatively straightforward. For example, if the component is polyethylene glycol, you just need to specify the molecular weight. For carboxymethylcellulose sodiums, a good practice is to specify the viscosity grades of these components. For ethylene vinyl acetate copolymers, you need to specify what is the vinyl acetate contents in the polymers. In addition to these straightforward cases, there are some other complex cases that may require additional information, sometimes comparative characterization from the test and R&D products. These examples including polylactide coglycolide, PLGA polymers, cross-linked hydrogel, as well as PDMS. I will talk about these examples in the later slides. Our first case is PLGA. PLGA is a biodegradable copolymers made of lactide and glycolates monomers. And PLGA has been used as the rate controlling excipients in more than 20 FDA-approved long-acting injectables and implantable drug products. The key polymer properties for PLGA, including its molecular weight and weight distributions, lactide to glycolide ratio, end groups, for example, whether it's acetic ends or it's ester ends, inherent viscosity, glass transition temperature, and polymer architecture. This refers to whether this polymer is a linear PLGA polymer or it's a branch polymer like the glucose co-star shaped PLGA shown here. It's important to know that the polymer properties can be changed during the manufacturing process of the drug product. So it's important that when you establish Q1s, you need to categorize what's the PLGA is actually in the finished drug products. Goduva research has been conducted to develop PLGA categorization methods, and this has been very helpful to support the Q1, Q2 evaluations. For instance, here, Goduva research has developed extraction methods and categorization protocols for PLGA analysis from finished drug products. And in addition, Goduva research has developed methods to categorize the glucose co-starch rate branch PLGA polymers. Goduva research also developed separation technique that can help to differentiate PLGAs based on different L2G ratio. For instance, some of the PLGA product may be made by more than one PLGA, so the separation method can be very helpful for reverse engineering. To support Q1 sameness of PLGA, an applicant should provide comparative categorization data. This data should be extracted using the PLGA polymer extract from the test and R&D products, including polymer composition, referred to the molar ratio between the glycolide and lactide, the molecular weight and weight distribution, as well as PLGA architecture. If a branch PLJ is used, the applicant should also provide branch analysis between the test products and RRD. For quality assessment purpose, the applicant should provide categorization on the PLJ, including but not limited to polymer end cap analysis, inherent viscosity, and glass transition temperatures. For those products use multiple PLJ, the comparative categorization can be performed using the PLJ mixture extract from the finished drug products. Although great progress has been made in the PLJ categorization, there is some remaining challenges. One of the challenges is that for some of the drug products, especially for ophthalmic route of administrations, there is very limited material for those products because they have extremely small size. For example, here, the Durista and Ozurdex, the products are less than 1 microgram. To address this type of analytical limitations, GDUFA research is currently being conducted under a contract with Akina. There will be in November, there will be a FDA CRCG workshop focusing on generic ophthalmic product developments. In this workshop, there will be a presentation about the updates of this contract. If you are interested in the presentations, please register to these workshops. Right now, this workshop has open for registration. The second case is cross-linked hydrogel. Hydrogels are three-dimensional polymers network with high water content. Usually, chemical cross-linking method can be used to create covalent bonds between the polymer chains. This can form stable hydrogel networks. Here is one of the examples of dexamethasone ophthalmic inserts. According to the drug labeling of these drug products, this is a PLJ-based hydrogel made of 4-amp polyethylene glycol, hydroxysuccinimide, glutarate, and trilysine acetates, and hydroxysuccinimide fluorescence, and other components. The hydroxysuccinimide ester can react with the primary amines in the trilysines, forming amide. Therefore, in the finished drug products, the cross-linked PLJ polymer conjugate with fluorescence is the actual components in the final products. To support the Q1 assessments of this product, the applicant should provide detailed descriptions on starting material, ratios, and concentration of all starting material, as well as cross-linking methods to generate the hydrogels. To support Q2 assessments, the applicant should provide information on the amount of each starting materials. If there is any difference in Q2, it may be justified by comparable mechanical properties. In addition, the applicant needs to ensure that the amount of each starting material does not raise any potential safety concerns. There is one FDA internal research focusing on this drug product to support the product-specific guidance. This research also provides support regarding the categorization methods for the Q1, Q2 sameness. If you are interested in the updates of this research, please attend the FDA CRCG workshops. There will be one presentation on this topic as well. The third case is PDMS polymer. PDMS are commonly used as drug reservoir and release controlling excipients in intrauterine system and vaginal rings. It's a cross-linked polymers formed by curing of silicon prepolymers or copolymers. There are several cross-linking methods available. In addition to that, the polymer kits may contain additives. One example of drug product using PDMS polymer is laminal gesture releasing intrauterine system. As you can see here, these drug products contains a reservoir that made of the mixture of laminal gesture and PDMS. The cylindrical shaped reservoir is covered by a semi-opaque PDMS-based membrane, which contain the PDMS and colloidal silica. There is one research project focusing on these drug products. Throughout the GDUFA research, we have developed scientific foundation to evaluate the PDMS compositional sameness. This research including development of analytical techniques to measure and categorize the cross-linking density of PDMS polymers, to understand how the PDMS cross-linking density affect drug release in intrauterine systems, to understand the impact of curing chemistry as well as other excipients on formulation attributes and in vitro performance of the product. To support the Q1, Q2 sameness, applicant should provide compositional information such as what's the PDMS chemistry you use, what type of curing chemistry you use, what's the degree of cross-linking. You need to also provide quantity of each components as well as dimensional information and mechanical properties to support the Q1, Q2 sameness assessments. At last, I would like to use this slide to conclude my presentations. In general, demonstrating compositional sameness of a polymeric excipient is straightforward, but we do see some complex cases exist. In this case, they do require additional information such as comparative categorization to support the sameness evaluations. GDUFA funded research has been developed analytical methods and scientific foundation necessary to support the sameness evaluation in these complex cases. With that, I would like to conclude my presentation. Our next speaker is Dr. Sami Nazel.
Right, Ben, thank you, and good morning, everyone. Just to clarify that my talk today is divided into two presentations. So I'll be covering the first half, and the second half will be covered by my good friend and colleague from OPQ, Dr. Jinhui Zhang. So in this presentation, we will be covering or discussing key considerations when developing the product-specific guidance for oral semaglutide. Now, before I proceed with my talk, just a quick disclaimer that anything that I present today or perhaps discuss during the question and answer session represents my personal opinion and should not be construed to represent the FDA's official views and policies. My presentation will be divided into three sections. So first, I'll give a very short background on oral semaglutide. And then the bulk of my talk will be about those key considerations when developing the PSG for oral semaglutide. And then I'll conclude this talk with a very brief overview of analytical challenges and some future thinking and directions. And this is where my colleague from OPQ will come in, and he will cover in much greater depth and detail on those analytical challenges. So a quick background on oral semaglutide. Now, semaglutide is the first and currently the only approved oral GLP-1 receptor agonist. It's indicated for glycemic control in adults with type 2 diabetes. Now, oral semaglutide comes in two different formulations. There is formulation R1, which was approved in 2019, comes in three strengths, 3, 7, and 14 milligram. And then more recently, there was a new formulation that was approved, which is an R2. It was approved in 2024, comes in three strengths, 1.5, 4, and 9 milligram. Now, both formulations, R1 and R2, they require the use of permeation enhancer, and that is SNAC, because of the poor oral absorption of semaglutide. Now, the key thing in this slide is to highlight that there was a product-specific guidance that was published in 2021 when the R1 formula was approved. But then with the approval of the R2 formula, there were revisions made to the PSG, and the revised PSG was published actually only a few days ago in October of 2025. So this presentation aims to tell you about 5 key revisions to the PSG from 2021 to the 2025. And what you see on this slide is a snapshot of the PSG page, and it is released. So if you can, it's available on the FDA website. So I encourage everyone to take a look at the detailed document. So the 5 key changes that I want to discuss today is number 1, expand study recommendations. Change number 2, we added language on API sameness and purity assessment. And change number 3, we clarified what do we mean by quantitative similarity. Change number 4, we added a language on washout period. And then change number 5, we added language on GI adverse effects and safety measures. So in the next few minutes, in the remaining slides, I'll be go over these 5 changes one by one and very briefly. So we'll start with the first revision to the PSG, and that is we expanded study recommendations to accommodate new strengths. And now before I go into this slide, just to emphasize that the PSG provides 2 options for applicants to pursue to establish bioequivalence. There's option 1 and option 2. In this slide, I'm only discussing option 1, which is basically we recommend BE studies on all strengths. So in 2021, when we only had the R1 formula, there were only 3 strengths, so we had 3 BE studies. But in 2025, now that we have formula R1, which is 3 strengths, and formula R2, which also has 3 strengths, so now we recommend 6 fasting BE studies. And that is single-dose studies on the higher strength and multi-dose studies on the lower strength, the 3 milligram and 1.5 milligram. So obviously the rationale for that is obvious, which is the approval of the R2 formula. But it's very important to emphasize here is that the formulation R1 and R2 are not identical. They are different, and these formulations are not substitutable on a milligram to milligram basis. And there is a difference in oral bioavailability between R1 and R2 formula. So that is the first revision or change to the PSG. The second change is that we added language on API sameness and purity assessment. And the language says that semaglutide can be produced using synthetic or semi-synthetic rDNA methods. Now, the reason we added this language is because we received a lot of inquiries from the industry asking a question of whether they can use recombinant DNA-sourced semaglutide in their ANDA submission. So basically, they're asking if it qualify for a 505J pathway. And the reason for these questions and perhaps confusion is because we have a guidance document we call the peptide guidance, and the detailed title of the guidance is at the bottom of this slide. I'm not going to go into the entire guidance. I just want to emphasize that guidance primarily emphasizes injectables. And we do recognize that the oral route of administration is generally associated with lower immunogenicity. And we also recognize that oral semaglutide, because of its low oral bioavailability, it presents a lower risk of immune response. So taking that into considerations, we came up with the language that it is possible to use peptide of both manufacturing process. So that 2025 PSG clarifies that it is feasible to use both synthetic and rDNA manufacturing methods in their ANDA submission, i.e. to pursue a 5,5J pathway. Now, revision number 3 is we added clarifying language of what do we mean by quantitative similarity. Now, this applies to option 2 in establishing bioequivalence. And option 2 says that an applicant may pursue a BE study on the highest strength, and they may request waiver for a lower strength. But in the 2021 PSG says that that option is feasible if the product is qualitatively the same and quantitatively similar to the RLD. However, there was no clarification or definition of what do we mean by quantitatively similar. So just a quick background is that when I say that something is qualitatively the same, it means that both products have the same inactive ingredients. And when I say that something is quantitatively the same, it means that those excipients between the test and the RLD are within plus minus 5%. Now, the question becomes is that when I say quantitatively similar, similar instead of same, what does that mean? And then we don't have an official definition of what do we mean by similar. So this is what we did in the 2025 guidance, is that we added clarifying language of how do we do an assessment of similarity. So we added the language saying that a test product is considered to be quantitatively similar if we apply, there are 2 criteria here. Number 1 is that the amount of SNAC is the critical absorption enhancer in the formulation, is within plus minus 10% of the amount of SNAC in the RLD. So that's criteria number 1. And then criteria number 2 is that the cumulative difference of all excipients calculated as percent of total tablet weight between the test and RLD is within plus minus 10%. So this is how we do the assessment of what do we mean by quantitative similarity. So obviously the rationale we added this to emphasize the importance of SNAC in the formula and also to provide this clarifying language of what do we mean by these terms. Now, change number 4 is we added a language on washout period recommendations. So the language that we added is, we said, and I'm going to read this from the slide, it says ensure an adequate washout period between treatments in the crossover study due to the long elimination half-life of semaglutide. Alternatively, a parallel study design may be considered. So we added this language because semaglutide is known to have a long half-life of about 1 week. And per FDA BE guidance, the general bioequivalence guidance, there's a recommendation to have a long washout period of about 5 times the drug's half-life. So we just wanted to emphasize that point in the PSG. Then in the final change that we introduced to the PSG is that we added language on GI adverse effect and safety measures. So again, I'm going to read the language that we added to the PSG. So the language says that due to the potential impact of semaglutide-associated GI adverse effect on subject dropout rates and study power, incorporation of safety measures in study design may be considered. Now, the reason why we added this language is because it is well documented that semaglutide is associated with GI adverse effect, primarily vomiting and nausea, and this adverse effects may lead to dropout. And so the concern is that if there is a high dropout rates in these BE studies, that it may compromise study validity and statistical power. So in applicants, they should consider that when they design their BE studies. So here are some thoughts on study design consideration when we are dealing with, in this case, GI adverse effect, that an applicant may consider perhaps a titration scheme in their BE studies. However, it should be understood that with titration, that it may increase study duration and will increase participant exposure to the drug. Alternatively, perhaps an applicant may consider using antiemetics to manage vomiting and reduce dropout rates. However, it should be understood that antiemetics may have an effect on gastric motility and drug absorption, and this should be taken into consideration. So the point here is that whatever approach that the applicant is using, it should be justifiable and should demonstrate that whatever design they're applying does not confound study results. So talking about this design consideration, I just want to point out that yesterday there was a fantastic talk by the clinical team from OGD on mitigation strategies in VE studies by Dr. Cannon Lee and Dr. Zoe Gwen. So I encourage everyone to review or take a look at that presentation. It goes into much greater detail in addressing these issues. It's a fantastic talk. So, and also another thing I need to point out is that obviously I only highlighted some of the five key changes, but the PSG has perhaps some old nuances and minor languages that I encourage everybody to look at the entire PSG for all the differences between the two, the 2021 and the 2025. So I'm going to conclude this presentation with a very quick overview of analytical challenges and future directions. First, there is an interest in using alternate absorption enhancers other than SNAC. So using different absorption enhancers raises regulatory questions on how to develop BE recommendations for those non-SNAC formulations, and there are also questions on how to create waiver pathways for products that use alternate absorption enhancers other than SNAC. And obviously, there is also a need for advanced analytical techniques because, for example, if we can improve the limit of quantification, perhaps we can pursue a single dose studies for the lower strength doses of 1.5 and 3 milligrams, and we can address perhaps SNAC matrix effect, and also improved method of analysis will enable us for more precise characterizations of peptide-related impurities and better API sameness assessment. So that really concludes my lecture. I just want to say that I acknowledge everyone who was involved in the development of the PSG, and there are a lot of individuals who are involved, and in particular, I want to acknowledge the MR team, the modified release team in the Office of Generic Drugs. Now, I do not have an acknowledgement slide because my colleague will have acknowledgement at the end of his presentation. So here I conclude my talk, and I thank you so much. And Jinwei, it's all yours. Thank you so much.
Hello, everyone. Thank you very much, Sammy, for the very nice presentation on the update of the product-specific guidance, which perfectly set the stage for me for this part. So in the next 15 to 20 minutes, I'm going to dig a little bit deeper into the science behind the rationale Sammy just talked about during his presentation. So you're going to hear me talk about his presentation a couple of times. So first, I'm going to talk something about the pharmaceutical development of semaglutide tablet. Then I'm going to talk about our in-house research work on impurity analysis and analytical challenges to echo what Sam just mentioned in the last part of his presentation. And my favorite part of my presentation is the question to be addressed, which means we still don't have answer. So please stay online, don't leave. Let's first talk about the pharmaceutical development, which happened between before the approval of semaglutide tablet. So you still remember Sammy mentioned about the bioavailability of oral semaglutide just below 2%, but less than 2% is still much, much greater than 0. It gave patient an option of oral administration, right? That's great. So you also hear Sammy mention about a couple times about the key excipients, which is SNAC, which enable the oral absorption of semaglutide. As a chemist, I'm going to blame Sammy forgot to put a structural snack in his presentation, which I put here. So you can see the rather very simple or straightforward, surfactant-like molecular. This key excipient snack enables the oral absorption of semaglutide. So current understanding of the absorption mechanism. So first, the absorption happened in stomach, not intestine. So if you look into the label, take the semaglutide tablet fasting, and with less than 4 ounces of water, which means just more than half cup of water. And after, eat or drink at least 30 minutes after you take the tablet. And the maximum absorption mainly is fall into 3 catalogs. First, the snack itself can enhance transcellular permeation of semaglutide itself. And also, as a surfactant-like molecule, it prevents semaglutide from forming oligomers. It's very straightforward. The smaller, the easier to be absorbed, right? And in addition, snack is weak base. It has a buffering effect in the gastric, which increase the local pH, prevent the activation of pepsin, thus prevent semaglutide being degraded by pepsin in the stomach. Collectively, these 3 mechanisms contribute to the oral absorption of semaglutide in stomach. And also, you heard Sammy talk about the GI side effect, and the delay of gastric emptying might be taken into consideration for clinical study. So the NDA holder, they got their original formulation approved in 2019, and 5 years after that, they got the formulation R2 approved, right? So they published their comparable bioavailability paper earlier this year. So you can see with almost half of the strengths, they are still equivalent. That means a lot. So taking together fast and synchronized release of both semaglutide and the snack is the key. Okay, challenges. So you heard about Sammy talk about challenges about the impurity, right? So there's specific issues with the tablet dosage form. Let's start with the basic control of drug substance and drug products impurity profile, right? And I think all the analytical chemists prefer the solution dosage form rather than tablet, right? Because solution can be analyzed as is. There's the issue of extraction efficiency and interference and the matrix effect from the excipient, right? And also for tablet, there's issue to be considered as discriminating capacity of dissolution method for in vitro B. So Sammy spent a lot of time talking about the Q1, Q2 sameness or similarity, right? So even so Q1, Q2 sameness means the manufacturer parameters might not be same, right? So the formulation R2 of the NDA holder is perfect example. When they use the same API, they use the same key excipients, but with almost half strength, they achieved the equivalent. So what are the critical processing parameters? And how those parameters can affect in vivo performance, which reflect by clinical B, and in vitro performance, which reflect by dissolution release test. And if non Q1, Q2 sameness, which using alternative excipients Sammy just mentioned, all the challenges for tablet still applied, and API excipients interaction might necessitate a different regulatory pathway. For example, if developer designs a formulation based on intestinal absorption, if you still follow the instruction for the ILD, let the tablet stay in the stomach, that's unfair, right? It can be easily degraded by pepsin in the stomach. So those are all the challenges. So next 3 slides, I'm going to talk about some in-house lab work. This is about impurities. And sorry for the busy slides. I mean, the reason is because ICH-Q14 guideline, which is analytical procedure development, and the ICH-Q2R2 guideline, which is validation of analytical procedure, just finalized a little bit less than 2 years ago. So in this slide, I'm just using a couple of different scenarios to highlight how Q14 and the Q2R2 guidelines can help the profiling and the quantitation of impurities in such a complex API, semaglutide. The whole concept of ICH-Q14 and the Q2R2 are knowledge and risk-based analytical procedure development and the fit-for-purpose analytical procedure validation. So for example, let's talk about impurities profiling, which is qualitative by nature. Semaglutide might represent the most difficult small molecule. It's still regulated under NDA, right? But the workflow pretty much need to take advantage of all the workflow for large molecules, though the detailed approach might be different. So high-resolution mass spec is the go-to method for impurity profiling. And we choose to do the top-down approach to avoid the necessary for sample processing and digestion. And using a high-resolution mass spec with different collision energy, collision mechanism might enable to the maximum coverage of sequence of all the impurities. And for potential matrix effect from tablet extraction, we use different approach such as spiking semaglutide standard and spiking internal standard to semaglutide tablet, which I'm going to go over, in the next slides. And analytical variables associated with study sample, we use standard to track matrix effect. We monitor the multiple ions to ensure specificity per ICH-Q2R2 recommendation. The last but not least, whether the data makes sense, that's very, very crucial. For example, if you develop semaglutide using a recombinant manufacturer, but you find a lot of addition and deletion impurity, which is pretty common in synthetic manufacturer. If I were the analyst, I need to go back to check if everything right. So here, example of impurity structure elucidation, which is formylhistine, which is the N-terminal amino acids of semaglutide. It's pretty standard high-resolution peptide mapping for impurity profiling. So the blue highlighted amino acids there, the B ions, which is identified, and the red there, the Y ions, which is identified. So I purposely choose this impurity because it's the N-terminal modification formed by the formaldehyde in the excipients with the semaglutide. So this slide shows the data about the evaluation extraction efficiency, which is very important for tablet dosage, for solid dosage form, and evaluating the matrix effect per ICH-Q2R2 recommendation. On the left side of the slides, it's extraction efficiency. We use the semaglutide neat solution and extract from tablet to compare their signal intensity. The red one is the standard and the green one is extracted. And the two figure, we are using 3 different ions. We were using high-resolution mass spec. We are using 3 different monoisotopic ions, which is based on ICH-Q2R2 recommendation. On the right side of the slides is evaluation the matrix effect. In this case, we are taking a little bit different approach. We directly spike the 8-deutery substituted semaglutide to the semaglutide, commercial available semaglutide tablet extraction. As you can see, the red was the neat standard, and the green was spiked into the tablet. We can see there's a minimal matrix effect affecting the quantitation. Let's move to the bioanalytical challenge to demonstrate bioequivalence. You heard Sammy talk about option 1 and option 2, right? For both options, you got to do the clinical bioequivalence for high strengths, right? For option 1, you got to do the bioequivalence clinical study for all the strengths, right? So the devil is in the details. Here, I want to emphasize that the earlier pharmaceutical development of semaglutide product might have been hampered by matrix interference in the incurred sample for a validated ligand binding method used for quantitating semaglutide in plasma matrix. I want to explain a little bit. During the earlier pharmaceutical development, NDA holder, they used the clinical study to compare the different combination of semaglutide and the SNAC. And then they take the human plasma for quantitation. And initially they developed a ligand binding based method. I mean, everything's fine, they validated. But when they applied the method for sample analysis, they encountered severe matrix interference. So in their publication, they document that the analytical data need to be interpreted cautiously. So that's the reason they switched to mass spec-based method in the later development stage. And there's numerous things need to be taken into consideration for bioanalytical method. I mean, here I just using one figure to show the importance of the right use of internal standard. If you are familiar with ICH-M10 guideline, which is a validation, bioanalytical method validation and sample analysis, for LC mass-based bioanalytical method, internal standard is a must-do, right? So in this case, we are using D8 semaglutide, which means 8 hydrogen in the semaglutide molecule was replaced by deuterium. The molecular weight difference is by 8. So for most of the small molecules, it's perfectly fine, right? Can be very well resolved. However, semaglutide is a molecular weight around 4,000. So it did not, I mean, its most abundant precursor ions were bear 3 or 4 charges. The ions bear 3 charges is very hard to fragment it. So typically we were using the ions bearing 4 charges. That means M/Z between semaglutide and the D8 semaglutide just 2. And for unit resolution mass spec, you can see the solid bar. If the resolution was set at wide mode, that means the D8 semaglutide channel will pick up semaglutide signal, which is really something we do not want. Again, devil is in the details. Thank you everyone for staying online. This is my favorite part. More questions to be addressed. That means a lot of things we still don't know. And first, is there any direct and specific interaction between SNAC and semaglutide? If you look at it in literature, academic and the industry, they investigated a lot of efforts studying the mechanism of interaction between SNAC and the semaglutide using different models. So the current understanding still think SNAC just serving as a chaperone for semaglutide to promote its absorption. But there's still some evidence indirectly point to that there might be some direct and specific interaction between SNAC and semaglutide. For example, you might still remember there's a hydroxy group on SNAC. Well, if the hydroxy group on the SNAC was moved a little bit, preclinical study indicated that the absorption of semaglutide will drastically reduced. Whether there's any specific interaction, we don't know. Thus, we and the whole community are working on. Also, you heard about in Sammy's presentation talking about both recombinant and synthetic semaglutide. It can be used for the end development, right? So recombinant semaglutide produced by yeast and translated in the yeast translation machinery. For synthetic, typically the current, the most common approach is solid phase synthetic, right? So for peptide with 31 amino acids, its higher order structure is something like a gray area. So whether there's any higher order structure difference between recombinant and the synthetic semaglutide, We don't know. There might not be big difference, but there might be some transient difference. And we are currently using a hydrogen deuterated exchange mass spec to investigate these two questions. If there are any potential risk, we're going to communicate with the community to de-risk upfront at the earliest possibility. So please stay tuned. Also, CFR allows the ANDA using different buffer and the pH. In most cases, it might not be a big deal. However, there's a publication talking about at more acidic pH, semaglutide tend to form aggregates and stressed and just shear stressed condition. So all these questions need to be answered. So I promise this is my last slide is talking about the two more questions. How to ensure the discriminating ability of dissolution method. So in option 2, dissolution is the to-go method to establish the in vitro bioavailability. If there's, as I just mentioned, the fast and synchronized release of both semaglutide and the SNAC is the key. If there's difference, we want the difference to be detected. If there's no difference, we do not want to see the artificial effect, right? And the last, should the clinical study design take into consideration of generic emptying deleting effect of semaglutide? There's an apple to orange comparison. I can use that to justify the question I'm presenting here. So if you look at the NDA holders paper, when they were developing the original formulation, single dose PK, okay, the patients all fasting overnight, and all the subjects were continuous fast 5 hours after the dose. However, look at the publication on the comparative bioavailability earlier this year, is steady state PK study. So patients still fasting overnight, but they were allowed to eat and drink after 30 minutes. Though it's still apple to orange, but it might indicate some things we need to take into consideration for clinical design. So with that, I'm going to conclude my presentation. Hope my presentation can help you to understand the Sammy's presentation better. And Sammy forgot to put an acknowledgement slide. I do, right? And Sammy's name there, right? It's a long list. I won't go over everything. So, I mean, there's many people contributed. Thank you. Oh, with that, I'm going to hand it to Dr. Maotong Zhou, Division Director at OPQ for next presentation.
Thank you. So I'm going to present the first case study on Glatiramer acetate injection, and my colleague, Dr. Kedu Chen, is going to present the second case study. So here's my outline. I first will give a brief introduction on this Glatiramer acetate. Then I'm going to talk about how to establish the API sameness for the Glatiramer acetate. And I'll list some common deficiencies, and I'll conclude the slide in about 10 minutes or so. So first introduction, Glatiramer acetate, the ROG is a Capsules, okay, Glatiramer acetate injection. Indication is multiple sclerosis. And the API is a mixture of copolymers of 4 amino acids, basically glutamic acid, lysine, and alanine, and tyrosine. Average molecular fraction of 0.141, 0.338, 0.3428, and 0.059. And average molecular weight is by 5 to 9 kilodalton. And although it's a mixture, but we still consider this as one single API. So here are the 4 amino acids that are used to manufacture the Glatiramer acid. Glutamic acid, lysine, alanine, and tyrosine. Of course, those amino acids are activated by N-carboxyl, anhydrate. So, and also the for glutamic acetate, the second carboxyl group are protected by benzoate, and also for lysine, the terminal amino group was protected by trifluoroacetate. And yeah, here is a synthetic skin I took from the J. Addison's paper. And of course, you have 4 activated amino acid. Then during the polymerization with the initiator of diethylamine in dioxane, then you do the polymerization. After the polymerization, you have the polymer, accrued polymer. The next step is depolymerization and also deprotection with the HBr and acetate, acetic acid. Then after you remove the first, first protection group from the Glatiramer acid, then the second deprotection with the piperidine to remove the trifluoroacetate group from the lysine group protection. The final step basically is purification. Use ultrapurification to remove all the smaller molecule impurities, also the reagents from the reaction mixture. Some key feature of the polymerization reaction is, we call it conserved characteristic of copolymer chain. So this polymerization is neither entirely conserved or nor completely random. Basically, the reaction are determined by 2 factors. One is the reaction kinetics, the concentration, and also reactivity of the polymer. Let me go back 1 slide. So the concentration of each amino acid, also the reactivity of the each amino acid. So the entire polymer chain, the composition of the 4 amino acids are not remain constant. Initially, for example, the smaller alanine, it's more reactive. This is more reactive and also have high concentration. So you can imagine that initially more of the alanine group will be incorporated into the polymer sequence. But later on, as the alanine are consumed, then you have less alanine available in the reaction mixture. Then the other components have become more available. So you have more of the other amino acid be incorporated into the polymer chain. So this phenomenon is called a propagational shift. Okay, that's one of the major characteristics of the Glatiramer acetate. So that's why we call it not entirely, random, also not entirely conserved. Second, because of this phenomenon, so there's not going to be a batch-to-batch, consistency, even you use exactly the same condition. So the sequential variation would be exist across the polymer chain. Then also, but locally, you still see some, conservation of the sequence of the amino acid. And like I said, Glatiramer acid is the API is a complex mixture of polymers, not a single small molecule. That's why we're requiring totality of evidence to approach it to demonstrate the API sameness. So why API sameness? For FDA to approve a drug, generic drug, the drug should be therapeutically equivalent to the reference drug product. They have to be bioequivalent, also pharmaceutical equivalent. And what is the pharmaceutical equivalent? Basically 3 criteria: same active ingredients, same API same dosage form, same route of administration, same labeling with some exception to produce the same clinical effect and also safety profile. And FDA cannot approve a drug without adequate demonstration of the API sameness. So for the Glatiramer acid, how to demonstrate API sameness, FDA published a guidance, PSG on this product. This PSG was recently revised just a few days ago, October 2025. So we have four criteria to demonstrate API sameness. First one is equivalence of fundamental reaction scheme. So you have to use the same, reagents, same amino acid, same initiator, which in this case is diethylamine, and also the same reagents for the cleavage. Then also same equivalent of physicochemical properties, such as amino acid content, molecular weight distribution, also spectral fingerprinting. Third, this is the critical one, the equivalence of structural signatures. So during the polymerization and the depolymerization stages, so because of the reaction characteristic, so generate a lot of we call the process signature or structural signature of those reactions. So the generic drug developer have to capture those and to develop their drug product. So the last one is just a confirmatory evidence of API sameness from biological assay results. And I'm going to just go through very quickly some common deficiencies we encountered during review of the generic drug product. The first one is inadequate ILD characterization. And especially when you do the ILD characterization, you have to link your characterization to the API sameness demonstration. So what kind of batch you select, how many batches you use, what testing you are using, and what kind of equivalence criteria you are using. Those also have to be clearly defined and demonstrate that those information are sufficient to demonstrate your API sameness. Second is insufficient process understanding, and also have to link to the API sameness again. So for example, in the polymerization stage, how the reaction is initiated and how the, for example, initiator is going to impact the polymerization, also the concentration, also how the reaction is the polymerization is a propagation stage, especially regarding the propagational shift, just as mentioned a few minutes ago. Those are factors that have to be understood, and especially have to link to the API sameness. Also, of course, the depolymerization and the purification. And recently, we received a couple of applications that there seems some confusion on the diafiltration step, the ultrafiltration steps. So that's why we revised the PSG to include this section in the PSG. Also, inadequate data on structural signature. I mentioned that because of the unique characteristic of the polymerization and the depolymerization reaction, there are a lot of process signatures or structural signature, should be impacted, should be identified and also characterized. But to properly identify those structures, so you have to understand what's going on with the reaction, understand what's the reaction mechanism. Then you identify the proper structural signature and characterize them. Last one I list as the improper negative control studies. And the negative control study basically is trying to challenge the method, analytical method, how sensitive they are, and also how specific they are. And for more information, you can you can refer to FDA response to the citizen petition, and it's very detailed technical. There are very detailed technical information. A lot of information is available. So one thing I want to focus today is the ultrafiltration or dial filtration step, UFDF step. So this step is used to purify the crude mixture after depolymerization step. The purpose is to remove the process impurities such as reagents, salts, and something like that. Also some impurities. But some lower molecular weight peptide components may also be removed, depending on the process conditions, such as the molecular weight cutoff of the membrane. So it is crucial to understand how this step, the ultrafiltration step, could impact the structural signature for API sameness. We see a lot of confusions. After some of the low molecular weight peptides are filled off, then how do you still demonstrate the API sameness under that condition? So here is an example from this publication. On the top, you can see on the top is size exclusion UPLC as trace. So you can see, when you use higher molecular weight cutoff for the membranes, then the average molecular weight will be higher. And if you are lower, then it's going to be lower. The same in the lower phase, in the lower diagram. The lower diagram uses a strong cation exchange UPLC as well. So it's clear to say that the ultrafiltration step is going to impact some of the factors, or, for example, the molecular weight. So let me just put the whole thing. So the within the FDA, we had quite a considerable deliberation and discussion and research on this issue. So this represents our current thinking on this issue. So the ultrafiltration step should not alter the structural signature of the individual peptides, because in this step, there's no chemical bonding formation. When should I stop? Still have time? Oh, okay. I'm sorry. Kind of 15. Sorry about it. Yeah. So since this step does not involve any chemical bond formation and breaking, so basically this step should not alter any structural signature of individual peptide molecules. That's for sure. But on the other hand, this step could impact structural signature characterization, because many of those characterizations are based on collective or aggregate analysis of a factor. For example, molecular weight. It's an average molecular weight. So of course, if some of the smaller molecules, smaller peptides are filled off, it's going to impact the characterization. And for example, also some of the ratios, amino acid, because the polymer sequences are not unique or not even. So if some of the more of the amino acid could be filled off. So that's why we said that this step could impact the structural signature characterization based on a collective analysis. So we advise in the guidance, we advise that the appropriate technique, such as fractionation studies, can be used to study the impact before you have adequate demonstration of the structure of the API sameness. So that's why we revised the PSG last month, and this section was added into the PSG. Here's my summary. So Glatiramer acetate is a complex mixture of copolymers, but although it's a complex mixture, but still it's considered one single API. Second, we use, because of the mixture, so we use the totality of evidence for API sameness demonstration. We do not rely upon one single, evidence, but a totality of evidence. Then also structural signature is a key. Understanding and characterization of the structural signature or process signature are key to demonstrate, one of the key factors to demonstrate API sameness. My take-home message is study, you need to understand the ultrafiltration step, and it's going to impact the API sameness demonstrations in your data, in your information. So you need to understand what's going on with ultrafiltering, all the process parameters, such as molecular weight cutoff, temperature, how many membrane you are using, and all those things, and how it's going to impact the structural signatures in your characterization to how does it impact the API sameness. Here are some references are listed here. Mostly the PSG and also the some other published paper. And now I turn to Dr. Chen. Yeah.
Thanks, Maltang. So, hello everyone. My name is Keduo Qian. Today, I will cover another case study on the generic drug development on Pentosan polysulfate sodium oral capsules. So the ROD Almiron is approved in September 1996, and it is indicated for the relief of bladder pain or discomfort associated with interstitial cystitis. The main complexity on this drug is reside on the API complexity. So the API is a semi-synthetically produced heparin-like macromolecule carbohydrate derivatives. It chemically and structurally resembles glycosamine glycans, and the average molecular weight is 4,000 to 6,000 Dalton. Based on the agency's internal research, which is published in the AAPS journal listed here, the structure of the PPIs is mainly a linear beta-1,4 xylose with some two-position substitution of the methyl glucuronic acid. There are also observations of the reducing ion aldehydes and pyridines, and there are observations of the three-position acetyl substitutions. So as Maltang just explained, to demonstrate generic drug therapeutically equivalent to the RLD, bioequivalence and pharmaceutical equivalence should be demonstrated, which includes the API sameness. So per FDA's guidance on the API sameness, in most cases, the complex mixtures are treated as a single API because it is not possible to distinguish each and every specific constituent component in the complex mixture, which is intended to furnish the pharmacological activities. And to facilitate the applicant research on developing the generic version of the PPS, FDA has published the drug-specific, product-specific guidance. There are several criteria that we recommend. the applicant to demonstrate the sameness. First is equivalence of the source of naturally occurring starting material. The second is the equivalence of the physicochemical properties, which include but not limited to molecular weight distribution, the sulfation degree, sodium content. And the third is the demonstration of the equivalence of the monosaccharide building block composition and the chain branching, which include but not limited to xylose units, the sulfation pattern, the glucuronic acid groups, the linkages, and the anomeric configurations. Also, the applicant is suggested to provide a comprehensive characterization of the impurity profiles. As the PhD recommended, the source of the naturally occurring starting material should be the same. The starting material used to manufacture the proposed PPIs should be the same to the that manufacture the drug substance of the RLD. So applicant should demonstrate the botanical raw material identity. And we recommend that the same plant species should be used so that the applicant can use a method such as the DNA barcoding. Also, you need to make sure that the same plant species of your choice should be used constantly during the life cycle of the product. Also, we want to clarify here that the plant is the botanical raw material, and the starting material is the extracted xylose, as we just mentioned, from the plant source. So to establish the API sameness, multiple characterization methods are applied. And here we have a list of the proper primary characterization methods that can be used, but you still have your choice of method. It's just some methods that can be used to establish the API sameness. For example, the NMR, proton NMR, carbon NMR, and 2D NMRs are generally used to establish the general structure sameness. Also, the sulfation pattern, the reducing end composition, and the glucuronic acid position and content, the linkages, and anomeric configurations can be explored by the NMR study. The ICP mass and IC are generally used for the compositional analysis and comparative study for the sodium and sulfate rate. Also, the combust and elementary analysis. The molecular weight distribution are studied by gel permeation chromatography, and the polydispersity should be compared between generic and the RLD. The monosaccharide building block composition and the polysaccharide chain mapping should be studied, and some chromatography methods are generally used. Also, there are some standard studies, Raman, FDIR spectral overlap, and UVVs, and also the specific optical rotations. So here are some observations of common deficiencies we found in assessing the applications, and hopefully it can help the applicant to improve the quality of your submission. As Maotang just mentioned, first is the inadequate information of the RLD. The PSG has recommended the 3 batches of generic should be compared with 3 batches of the RLD sample, and it is to the applicant's advantage to test more RLD samples. Also, if you provide equivalence criteria based on any statistical approach, the justification or rationale should be provided for us assessment. The second is the insufficient process understandings. Please know that the manufacturing process is the critical determinant of the complex API identity, and the structure signature or process signature are molecular fingerprints left by the manufacturing process. So the process signature are the essential tools to characterize the API identity and quality. So these are related to the third observation that there are a lot of inappropriate classification of minor components versus impurities. For example, as we just mentioned in the structure of the PPS, the O acetylation and also the reducing end aldehyde and reducing end pyridines are really left because of the manufacturing process. And they are currently treated as the minor components, so they should be comparable to the RLD instead of being treated as impurities. And also there are inadequate data on structure signatures. For example, proper identity and characterization should be provided for all the NMR peaks corresponds to the structure signatures of the RLD. But on the other hand, if there are peaks that do not exist or has difference, they should be properly justified or explained. So here are some interesting research. This one is published by Ben Pharmachem. Even though there is no generic PPS approved in the US market, there are some mimics on the European and Asian market. And Ben has acquired those products and compared with the RLD. And they found that those mimics do not have the comparable xylose content in comparison with the RLD, which is shown in item B. On the other hand, there are also some interesting findings that listed in item C that the other chemical additional monosaccharide content, for example, the glucose, mannose, arabinose, ramenose, and galactose are quite different between the RLD batches and the mimics in Europe and the Asian market. So because of the process to produce Pentosan polysulfate sodium, we know that all the carbohydrates are come from the starting materials that are extracted from the plant. So we can see that a better understanding of the manufacturing process, which includes the understanding of the extraction process, can truly help the applicant to develop the APIs that are same to the RLD. Because right now it looks like the starting materials may have some differences. And here are a publication from the agency that it indicates the presence of the O-acetyl group of the pyridine rings at the reducing end, the anomeric configuration, and also the aldehyde. So all these are the minor components as I just clarified. And here are their content in the NMR study. So to summarize my talk, a PPS is a semi-synthetic heparin-like polysaccharide, and the API sameness is again assessed based on the totality of evidence, and also the structure signatures are the process of the residue. So we really, so we really put attention to those. So here are the resources that covers the PPS that for your reference. So I will pass the floor to Dr. Dahui Liu on ion products.
Thanks, Kedong. Hi, everyone. So now I'm going to talk about the comparative physicochemical characterization of iron colloid product. So at the beginning, I'm going to briefly have an overview of iron colloid products, and then I'm going to go to the physicochemical characterization to support the equivalence of generic and the ILD. And then I'm going to give several examples of the common deficiencies we see and our recommendations towards how to resolve those issues. And then I'm going to end with some closing thoughts. So iron colloid products are injection dosage forms widely used for the treatment of iron deficiency anemia.
The nano-sized complexes comprise of polynuclear iron core and the carbohydrates. The carbohydrates surrounding the iron core facilitate the dispersion of the nanoparticles in the aqueous formulation, and upon dosing, minimize the release of iron directly to the system, thus reducing toxicity. So here I have two proposed structure for the iron product. On the left is ferric carboxyl maltose. It is more of the conventional structure, proposed structure. It has a polynuclear iron core and is covered by the carbohydrate shell. On the right is a proposed structure for ferric maltose. It's a little bit different from the core shell structure. Instead, the iron core and the carbohydrate, they are associated to one another in this kind of like matrix pattern fashion. Currently, in the U.S. market, there's 6 iron colloid products. The first one is iron dextran, which was approved in 1974. The most recent one is ferric maltose. It's approved 5 years ago. In 2011, we approved the first iron colloid product is the sodium ferric gluconate. 10 years later, a generic ferromoxitol was approved in 2021. Most recently, in this year, August, we approved 3 generic iron sucrose. And if you look at the PSG column, the product-specific guidance, since 2012, the agency published PSG. Since then, there's more PSG being added and being revised multiple times. So this table, I cannot arrange the product by the size of the carbohydrate. The top 2, the sodium ferric gluconate iron sucrose, you can see the carbohydrates, they are small molecules, the gluconate, sucrose. The next one is ferric maltose. Maltose is oligomer of linear glucose with a repeating unit about 5. The 3 at the bottom, they are just different type of dextran. Because of the nature of the carbohydrates, such as the functional groups, the size, the association of the carbohydrate and the iron core, the strength differs. And that can be demonstrated in the strength, the iron load, or sometimes called that. And you can see those iron colloids with simple molecule carbohydrate, the strength is relatively low in this group. Sodium ferric gluconate is only 12.5 milligram iron per mil. Iron sucrose is 20. And with a bigger ligand, bigger carbohydrate, you can see it can go up to 100 for the ferric maltose, like 50 milligram per mil for the carboxyl maltose. Also in this table, I list the MDD, maximum daily dose, that is based on the labeling. This is actually depends on many factors, not only on the stability of the iron colloid particle. For example, for the iron dextran, the iron load is pretty high. It's 50 milligram per mil. However, the MDD is only 100. And that is mainly due to the anaphylaxis issue of the dextran. So as I mentioned, the agency published PSG for all the iron colloid products, and the structure of those PSG are pretty much the same. First of all, the test product has to be Q1 and Q2, the same as the ILD, because they all injection dosage. So the PSG recommend to be studies in vivo and in vitro, and also has this recommendation to demonstrate comparable physicochemical properties of the test product and the ILD, and that's the main focus of my talk. So also in this PSGs, it's very clear spelled out the requirement of these batches, test batches used for the characterization. The 3 batches of the test product has to be made using the commercial process. And that at least one batch need to be produced as the commercial scale, and that batch need to be used in the physicochemical characterization, as well as the in vitro and the in vivo B study. Now let's look at the complexity and challenges of the iron colloid product in terms of the characterization. I think this kind of shares similar challenges as the previous examples. First of all, when we look at the product is a mixture. Actually, it's a mixture of the colloid particle, free iron, unbound carbohydrate, and some other small molecular weight species. Next is nanoparticles, the complex. The iron core, and sometimes if the carbohydrate is a large molecule, they all have distribution of size, structure, and composition. So because of that, the common analytical methods for pharmaceutical products may not be appropriate for the characterization studies. And also because many of those studies and the sample preparation, sample manipulation is inevitable. However, those preparation may impact the actual properties of the drug product. So how do we deal with those challenges? I think for people who work on the iron colloid product are very familiar with this paper in 2017, the AAPS paper. In this paper, it lay out the map of the physicochemical characterization. It's a pretty comprehensive overview, gave many recommendations of many methods. So this table is very busy, but I want just make 2 points. The first is if you look at the right column, the common test, and you can notice many of those characterization methods are not well established in the pharmaceutical industry. Second point is, even though there's so many tests, so many technology are going to be used, we can roughly put them in 3 categories. That is the whole particle, the iron core, and the carbohydrates. So here are some of the considerations for the characterization study. As I mentioned, there's so many of those test methods are not well established in pharmaceutical industry. Not all of them are quantitative. In fact, many of them are qualitative or semi-quantitative. So because of this, that reason, we can expect it when we get the data set, you will face many uncertainty. At that situation, we recommend to use orthogonal method. So if the test product and RD are comparable, and when we use different orthogonal methods, has different principle. When we do the characterization from different perspectives, the test product and RD should behave the same. Again, the second I want to point out is pay attention to the sample preparation. The sample preparation should be consistent with the purpose of the test. For example, if the test is characterized the iron core, if the small molecule will interfere with the signal, then these small molecule species need to be removed by dialysis or ultrafiltration. The last point consideration I want to mention is, since the sample manipulation is inevitable, and that will impact the property of the product. So sometimes we suggest to conduct the study under a series of controlled varied experimental conditions to enhance the differentiation ability of the test. And I'm going to give an example later for that point. So now I'm just get into the case studies that will to give more to articulate kind of more on those considerations I just mentioned. The first example I want to give is the characterization of stoichiometry. The common deficiency we see kind of related because those drug products, as I mentioned, is a mixture of not only the iron colloid product and colloid particle, but also unbonded carbohydrate and free iron and other components. So to get a full picture of the stoichiometry composition of a drug product, we recommend to do the study, use dialyzed sample and also undialyzed sample. Another common issue we see that the provided data are limited is only the sponsor only provided the assay data of iron and the carbohydrate. Again, that will not give us the full picture of the stoichiometry picture. We recommend to provide data of other species like the unbound iron, carbohydrate, and other small molecular weight species. The second case study example is molecular weight determined by size exclusion chromatography. SEC method is a very commonly used method. It can be easily validated, so it's a very useful method for the characterization. However, we know when we do SEC analysis, the data is generated. It needs to be based on the standard curve generated for that set of experiments. So you can imagine, because the differences of the instrument, even reference standard, and the testing time, all of this will impact on the data, on the result. So we see this commonly when the sponsor provided data of the test and ILD, they've been tested at all kinds of different time. So it is hard to compare those data because you do not know the differences. Are they just from the different testing time, different testing condition? What's the real difference? So our recommendation is for the SEC study, we recommend to carry out those studies side by side. What I mean by side by side is those tests being tested at the same time, use the same instrument, the same lot of reference standard. Another thing we see commonly is the sponsor will use the USP monograph limit to justify the differences they observed in the study. If you look at the USP monograph limit for molecular weight by SEC of iron sucrose, it's a very large range. It is from 30-some thousand to 60,000. So as evaluation of those data, the USP monograph limit is generally considered as a minimum requirement for quality control. Therefore, they're not sufficient justification for comparability. Next, the example is on the sample preparation. So this is Moss Bauer spectroscopy is very useful tool for the iron colloid product. It is a very sensitive method, luckily because of the iron 57. So this method can be used to characterize iron core crystallinity, magnetic property, chemical environment, and also using the blocking temperature, the core size can be estimated. Another advantage of the mouse viral spectroscopy is it can be conducted using the as-is drug product formulation. So the sample manipulation is minimum. Actually, this is one of few methods that's require very minimum sample preparation. Many times we see the sponsor will provide mouse virus data. However, the sample is dried by just drying or by lyophilization. So those manipulation actually impact the property, and the data gets impacted. So our recommendation to use a mouse virus spectroscopy, because it's such a good tool, we recommend to use as-is drug product formulation for study. That's the first thing. The second, next thing, we also recommend to conduct the study at a series of temperature, using this spectra at a different temperature where the blocking temperature can be determined. Then I'm going to go to my last example. Again, this is for the sample preparation as well. So this is particle size distribution determined by dynamic laser scattering. Again, DLS method is a very commonly used method, so it's a very good tool. We like to have those data. However, using DLS study, the sample has to be diluted. So the dilution will kind of disrupt the nanoparticle, but that's exactly my point here. So our recommendation is to conduct the study at a zero dilution, and then we can see the behavior of the test product and the IOD, how they act under the zero dilution, and that data actually can be used to monitor to detect the interaction strengths between the carbohydrate and the iron core. With that, I want to end with some closing thoughts. So because of the complexity of those iron colloid products, so the evaluation of the equivalence do warrant a comprehensive approach. And you heard this a lot today. So the recommended in vitro BE study and in vivo study and the comparative characterization, the PSG, we see them as a different part of the comprehensive approach. They are complementary to each other, and they are not there to replace one another. So we consider them as a part of a totality of the evidence approach to demonstrate of the sameness. Again, the last point I want to emphasize again is the suitability of the characterization method, including sample preparation and testing condition. Those are very critical for the valid comparability evaluation. I have the acknowledgement slide here. This is an effort between the team, lots of team in the OPQ assessment side and OPQ research, and also from the AGDD. And thank you all very much.
Thank you to our speakers for those excellent presentations. We'll now take a short morning break and return at 11:05 a.m. Eastern Time for our Session 3 Q&A panel discussion. During the break, we encourage you to continue submitting your questions through the email at orsevents@fda.hhs.gov with the subject line of AGDD 2025, and we'll address as many questions as possible when we return.